Thank you, and I want to thank the organizers for giving me the opportunity to present some research to you today. The research I will be sharing with you looked at the importance of the modification of cysteine 144 on TRIM72 in regulating its known protective effects in ischemia-reperfusion injury. Okay, so just quickly, our lab has previously found that nitric oxide donors such as GSNO are cardioprotective, and that this cardioprotection is associated in this case and with, for example, preconditioning with an increased S-nitrosylation and many proteins, one of which is TRIM72. So let me tell you what TRIM72 is. TRIM72 is a protein that's expressed in the muscle, and it is known to have a number of functions, one of which is that it contributes to muscle membrane repair during ischemia-reperfusion injury. TRIM72 has also been reported more recently to impair insulin sensitivity. And just quickly, I want to mention that TRIM72 is also referred to in the literature as MG53 or Mitsugumin 53. Okay. So previous proteomic experiments in our lab identified S-nitrosylation of TRIM72 at the residue cysteine 144. And interestingly, during preconditioning, which protects the heart against ischemic injury, there is actually a dramatic rise in the amount of TRIM72 that is S-nitrosylated at cysteine 144. And this suggested that S-nitrosylation of TRIM72 might contribute to this cardioprotection. Okay, so interestingly, in follow-up experiments, they found that the amount of TRIM72 protein in the heart decreased during ischemia-reperfusion. However, under conditions of elevated S-nitrosylation, this decline in cardiac TRIM72 was blunted. And notably, GSNO reduced infarct size. Okay, so the overall idea is that normally oxidation at cysteine 144 is expected to cause a decrease in TRIM72 protein levels in the heart. However, if cysteine 144 is S-nitrosylated, the residue cannot be oxidized, which thereby prevents the decrease in TRIM72 protein and reduces cardiac injury. So this finding prompted us to generate a mouse with the cysteine 144 on TRIM72 mutated to a serine, with the idea that the cysteine 144 to serine mutation would work similarly to S-nitrosylation by preventing oxidation at this residue. And we expected that these knock-in mice would be protected during cardiac ischemia. So to test this, hearts were Langendorf perfused and then subjected to 20 minutes of ischemia followed by 90 minutes of reperfusion. So this graph on the left shows the percent recovery of cardiac function at the end of the 90 minutes of reperfusion period compared to the pre-ischemic function. And as you can see, the knock-in heart has better recovery of cardiac function than the wild type hearts. In addition, infarct size shown here on the right was less in the knock-in hearts compared to the wild type mouse hearts, indicating less cell death. And as you might expect, when we looked along with our collaborators at Johns Hopkins, including Mark Kor's lab, we saw a similar protection in vivo. So in this experiment, the mice were subjected to an LAD occlusion protocol. After 45 minutes of occlusion, hearts were then reperfused for 24 hours. We saw better cardiac function and less injury, as indicated here, by a higher ejection fraction and smaller infarct size in the knock-in mice. So while we were trying to understand how the knock-in mouse is protected, this manuscript was published by Ru Ping's group, and they showed that TRIM72 can be released by muscle and that this is a process that is regulated And specifically, they showed that its release can be regulated by glucose and by insulin. So this suggested to us that perhaps the release of TRIM72 could be reduced in our TRIM72 knock-in hearts. And to start to answer this question, we again subjected a set of hearts to 20 minutes of ischemia and then collected the perfusate from the hearts during the first 10 minutes of reperfusion. And we found that TRIM72 release during reperfusion was much higher in the wild type compared to the knock-in hearts. And this wasn't just because less protein was being released from the heart, because due to less injury, because we did not see a difference in the amount of total protein in the heart perfusate. Okay, so this data might suggest that by reducing the amount of TRIM72 released from hearts in response to ischemia reperfusion injury, more TRIM72 is then available in the knock-in hearts for membrane repair, resulting in the lower cardiac injury we observed. Okay, so not only did Ruping's group report that TRIM72 secretion can be a regulated process, they also showed evidence that reduced TRIM72 in the circulation can actually improve whole body insulin tolerance. And in fact, this idea that TRIM72 regulates insulin sensitivity is not a new idea, because this group had also shown a few years ago that if you overexpress TRIM72, or also referred to as NG53, in mice, what you see is that these mice develop insulin resistance. Here you can see worse glucose tolerance and worse insulin tolerance in the mice with overexpression. Okay, so interestingly, this might suggest that our TRIM72 knock-in mouse might have altered insulin sensitivity. And to test this, we examined the insulin sensitivity of our wild-type and TRIM72 knock-in mice using a whole-body glucose tolerance and insulin tolerance test. And as you can see, the mutation of TRIM72 did not impair glucose or insulin sensitivity. And this indicates that this mutation was able to, interestingly, lead to cardiac protection without actually impairing or affecting insulin sensitivity. And we also think and are planning to test whether if we were to look at insulin sensitivity under conditions where TRIM72 secretion is elevated in wild-type mice, we might then actually see an improvement in insulin tolerance in our TRIM72 knock-in mice. Okay, so together these results indicate that modification of cysteine 144 may in fact regulate TRIM72, and specifically our data show that TRIM72 knock-in protects the heart against ischemic injury, and we found that there is more cardiac release of TRIM72 from the heart during reperfusion in the wild type versus the TRIM72 knock-in hearts. Okay, so this research was done with my mentor, Tish Murphy, and these members of the lab played important roles. And this was also a collaboration with researchers at Johns Hopkins, especially Mark Kor's group. Thank you.
So the first question is for Deepshika from Gary Loper's chat. Nice talk. How do you think BCKA are modifying phosphorylation of AKT? And then the follow-up question, are they interacting directly with AKT?
Thank you, Gary, for the question. We did see that BCKs could directly activate mTOR signaling. So we think that that is one of the way that it is modifying AKT phosphorylation. And also we found that BCKs can inhibit insulin-induced PP2A inhibition. So that is also another method that we feel. And as it is known, and we have also found that BCKs can directly impact mitochondrial respiration and increase ROS. so which can also influence AKT phosphorylation. So right now, actually, we have a couple of theories surrounding this.
The next question we have is from an anonymous attendee. They're talking about the fibroblast cell cultures. They ask, is cluster 8 a non-fibroblast cell culture the most deviating one, is what they mention. So I believe this question is for Alexandra Garvin.
Yes. Thank you for that question. We were worried about the same thing when we first saw the clusters. However, none of our cells express markers of either endothelial cells or smooth muscle cells or even hematopoietic cells. And so we know that these are fibroblasts. However, they are just a very unique subset, as is cluster 7. And one theory we have is that potentially these fibroblasts may potentially expand in different either injury states or other ways, but we're certain that those are fibroblasts. Why they're so unique, we're not incredibly sure.
Okay, so the next question is from Julie. It's a technical question for Deepshika. How did you measure BCAA level, whether you use a mass spectroscopy?
Yes, we indeed used UPLC MSMS for the intracellular as well as the serum and plasma BCA and BCKA levels, yeah.
Then we're going to jump back to Alexandra. The next question is, did you do spatial selection of the fibroblasts you subjected to RNA sequencing?
I'm sorry, I'm not exactly sure what you mean by spatial selection. If you mean localization to the heart, we just did a complete LV digestion and isolated all fibroblasts from the left ventricle. I'm sorry if that doesn't answer your question, but.
Okay, so the next question is also for you, Alessandra, from Sina. Is it possible to distinguish which portion of the heart each cluster of fibroblast comes from, that is closer to the epicardium versus closer to the endocardium?
As of right now, our analysis does not distinguish that, but some follow-up studies that we will be doing is some imaging in the heart and staining with some markers of these clusters in the left ventricle to see if potentially there are special localization for some of these clusters. And I'm very interested in that, actually, to know if some of these phenotypes are coming from different areas of the heart. So hopefully that is to come, but right now we don't know.
The next question is also for you, Alexandra. They ask, did you compare these fibroblast clusters to a non-hypertensive rat ventricle fibroblasts? And if so, which clusters are specific for hypertension?
We did not compare these to non-hypertensive hearts. The main question for the study was within the hypertensive heart, what the impact of ACE inhibition was. And so we did not have that non-hypertensive group in this study, and we haven't done that.
Okay, so the next question is also for Alessandra from Leslie Kennedy. So I think it's similar question what you just answered, but still I will just read out. Is there any evidence of fibroblast in differing region of ventricular myocardia having distinct gene expression? In other words, do you think the cluster group in cardiac region, or do you think the clusters may be more heterogeneous, spread throughout the myocardium? This may impact crosstalk between fibroblasts and cardiomyocytes in the left ventricle versus those are in the right ventricle.
Yes, great question. And I agree with you that potentially different localization of these clusters would impact the crosstalk. And although right now we don't know, I wouldn't be surprised if we find some of these clusters are in particular areas. I know that there's some evidence out there to suggest that fibroblasts from either the endo or the epicardium serve slightly different roles. And I'm eager to know the question to this, or the answer to this question.
Great. The next question is, how do you isolate the LV fibroblasts for single-cell RNA sequencing?
Sure. So after we isolate the left ventricle, we mince up the tissue and put it through a couple rounds of digestion with an enzyme called liberase. And the fibroblasts that are released from that digestion are then plated, and since fibroblasts are so adherent, we use those adherent collagen producing cells, which is what we used for this experiment.
Alexandra, may I just interject? So maybe they mean, which methodology do you use to isolate cells? Do you use Langendorf, non-Langendorf methodology?
Oh, so we don't cannulate the hearts, and we don't digest the heart when it's in its full form. I know some people will perfuse an enzyme, but what we do is we just mince up the tissue, after it's removed, and then from that minced up tissue, we submerge it in the liberase buffer and put it through several digestions and collect the supernatant, and then let the cells adhere, if that clarifies.
Okay, so next question is from Ron. He's asking two questions. Were the RNA sequence experiments from the hearts at a time point with overt tissue fibrosis? And the next question is, any plans to build a progressive data set over the progression from hypertension to HT to dysfunction?
Yes. So at this point, the hearts are certainly hypertrophied, and there is an amount of fibrosis, but it's not overt. I would not call it overt fibrosis. If you follow up with other injurious stimuli, you can stimulate higher amounts of fibrosis in these hearts at this time. And as for the second question, any plans to build a progressive data set? We would love to with RNA-seq, and it is, I think, an overall goal to understand the timeline of these changes, but right now we're focusing on how responsive this cell type is to our treatment that you'll hear about on Friday.
Oh, sorry, I'm talking when I'm muted. The next question's for you, Alexandra. From Mohan, they ask, could you please elaborate on the enzyme cocktail used for single cell preparation?
I think I already answered that. If you mean for cell isolation, we use liberase as our enzyme.
Okay, so the next question is also for you, Alessandra. This is from Jun Sun. He's asking, besides co-treatment with ACE inhibitor enalapril in angiotensin II induced hypertension, have you tried to treat the rats with nitric oxide donor? If so, is there any difference in cell cluster between angiotensin II plus enalapril and angiotensin II plus NO donor?
So good question. We haven't done those exact experiments, but previous experiments in our lab have utilized a NOS inhibitor. So not a donor, but in our hypertensive hearts that either got the transient ACE inhibition or not. They also then followed up with an L-name NOS inhibitor. And in those studies, isolating the cardiac fibroblasts and then looking at them in vitro, our transient treatment does protect against some of the impacts of that L-name treatment in terms of proliferation and collagen gene expression. So we know that this transient treatment does play a role in protecting against some of those pathways, but in terms of the single cell sequencing, we haven't done those experiments.
All right, the next question, and maybe if individuals would like further or additional information, they can feel free to reach out to the individual speakers if they feel like the answers weren't adequate to kind of what they're asking for. But the next question to Alexandra as well is, while classifying the 9 subpopulations into 3 classes, what were the criteria being used to group them into 3? For instance, does it mean the subpopulations of fibroblasts classified under oxidative stress are programmed to express different kinds of oxidative stress response genes?
So the way that I went about, going through this data is we have thousands of different differentially expressed genes, and what we were looking for were, I chose these three categories because of the roles that they play in fibroblast activation. And so in terms of actually grouping a cluster into one of those areas, that's a little bit harder to do, but more just looking at overall, was this cluster more proliferative or more inflammatory or more fibrotic, was a way for us to help understand potential contributions of these clusters to overall, cardiac fibrosis. And so I wouldn't say that we've necessarily pinned them down to be one or the other, but just over all of the genes that were, uniquely expressed in a cluster, we pulled out these processes because they were relevant to our questions and because they were areas where we saw some unique characteristics between the clusters, just to help understand their contributions.
So Alexander, Richard is wondering, what are the markers did you use to identify your fibroblast population after isolation?
Markers in terms of purity, I think, is what they're asking. So ultimately, we know that all of the cells that we isolated express things like vimentin, and that they don't express markers of other cell types that we may potentially get in this prep, such as vascular smooth muscle cells or endothelial cells or immune cells. We didn't see CD68 or CD31, smooth-lin, any of those markers of potential contaminating cells in the fibroblast prep. So we're confident that we're looking at mainly, collagen-producing fibroblast cells.
All right, the next question from George is, do you know if the change in fibroblast cluster morphology in response to hypertension is terminally differentiated or reversible? And then there's a second question. Well, I'll read them both, then you can answer them both. And the second question is, do you know whether female mice undergo the same transformation in fibroblast differentiation in respect to insulin, given the sex-specific differences in collagen deposition?
Okay, great question. So the first one, we don't know in terms of, differentiation, whether these cell, states may be reversed either with a treatment or, if they are reversible at all, or if potentially these cells might, die out and be repopulated. We don't know if these are reversible phenotypes. But all interesting questions in how, a targeted treatment may impact these clusters. And then the second question, we have not done any studies with females, but of course that would be in the future directions, and knowing how different they are and how relatively protected a female heart is, it would be interesting to see if these same clusters would come up.
Okay, so now Monica is asking, how I missed it. Okay, so how you isolate pure population of cardiac fibroblast from the heart. Then the second part of her question is also, it seems that you have 4 distinct population of fibroblast. Wouldn't you expect to have more visible transition from one population to another?
So I think I've, hopefully you've heard my answer to how we isolate the fibroblasts. So I'll, in the interest of time, move on to the second part. So we get 9 distinct clusters. I focused in on 4 clusters today, given their unique characteristics in areas of proliferation and fibrosis and inflammation. And so whether I would expect to see more visible transition from one population to another, if you're referring to the UMAP plot, where it's basically a cloud of cells with varying colors, I think that has to do with the fact that we're using a very pure population here. They were, rested in, culture dishes for 3 days, so they were adherent. they were growing for 3 days. This is a very pure population, so we don't get huge changes in gene expression like you might even see with different cell types in the heart when people isolate all the cells from a heart. So because these are, cardiac fibroblasts that were cultured for 3 days and then subjected to single cell RNA-seq, the fact that we even saw distinctions was exciting, and even more so the fact that we see an impact of our treatment, again, which you'll hear more about on Friday, was even more exciting. So I can't say that we would have expected to see a more visible transition given the fact that we were working with such a purified population of cells.
Great, thank you. We're going to switch over to Natasha now. Natasha, the question for you is, would C144S alter the ubiquitin ligase activity of TRIM72?
Okay, thanks actually for that question. So that was one of the first things that I actually looked at in hearts from these mice, because its ubiquitin ligase activity is believed to affect IRS1, cause its degradation. So I tried to look at overall ubiquitination in the hearts, and I didn't see a change in that, and I didn't see a change in targets of TRIM72 that are expected to be changed if its activity was changed. Having said that, I did not specifically run an assay looking at TRIM72's ubiquitin ligase activity. So if that answers the question.
So the next attendees is asking or wondering whether the effect you see is the enzymatic dependent protection or structural.
So sorry, I'm not specifically sure what that means, but what I'll say is I didn't see evidence of enzymatically affecting its enzymatic activity in terms of ubiquitin ligase activity. But other than that, I don't know the answer to that question.
All right, we're going to switch back over to Alexandria, and I believe you've already asked or answered this question, but just to make sure it's adequately addressed. The question is, did you find any differences in the cluster depending on the location in the heart?
Right, and unfortunately we don't know. We isolated fibroblasts from the heart altogether, and so unfortunately we don't know where each of these clusters are coming from, if they are specifically located, but something we plan to follow up on with some candidate markers using some imaging of the left ventricle.
Let's go back to our first speaker, Deepshika. So Azrul is asking, what is the glycogen content? The second question is, does it affect Krebs cycle intermediates?
Thank you for this question. We did not measure the glycogen content, but there has been studies, a recent study actually in a PPM1K phosphatase knockout mice, where they did find differences in glycogen metabolic intermediates, and they also incubated HepG2 cells, liver cells, with BCKAs, and they found an induction in glycogen phosphorylase mRNA level, which is responsible for glycogen breakdown. But it will be interesting to see what happens within the cardiomyocytes. For the second question, we are in the process of developing a method to measure the TCA cycle intermediates, but then again, previous studies have shown that if the BCK oxidation is inhibited, that results in reduced TCA cycle intermediate. So we are expecting a similar result with our study as well.
Thank you. All right, we're going to come back to Natasha. Was there a sex difference in the ischemia reperfusion results, and are females also cardioprotected?
So we didn't specifically compare male versus female TRIM72 knock-in mice in the ischemia reperfusion experiments. But having said that, we had a couple of female mice that were done, and they also showed some protection compared to the wild type mice.
So the next question is from Emma, and it's for Alessandra. I think the Srinivas part is you already answered, so I'm directly going to the second part of her question is, have you compared your single cell data and cluster with other model, such as the more ischemic model by Gladka et al. 2018?
Yeah, thanks for that question. And I know that paper you are speaking of, and if I recall, they looked in all cell types of the heart, cardiomyocytes and fibroblasts, and that they did find very distinct clusters. They found fewer clusters than we did, but very distinct ones. And what I have found in general is when comparing the clusters that we have to those from models of ischemia, post-MI, that those clusters tend to have much more extreme expression of markers such as, periostin and, these other markers that are really highly upregulated after an injury that we're not necessarily seeing differentially expressed. They're expressed in our cells, but not in not very highly, and not in distinctive clusters. And so in terms of comparing our data sets, I think it's relatively difficult given the methodological differences, but also I think ischemic hearts might show more extreme expression of some of those markers that we're not necessarily getting in our hypertrophied heart. I hope that answers your question.
The next question's from Svem and Alexandria. They're wondering if you compared your hypertrophy data set with the single cell data from mice after MI published by Richard Harvey's lab. And is there a specific cluster in both data sets contributing to cardiac fibrosis and scarring?
So thanks for that. And I think my answer will be similar to my previous one in that I can't say I've compared it to the specific data set, but to MI data sets in general, those data sets tend to have more extreme expression of activated cell markers. But whether there's a specific cluster in the data sets contributing to cardiac fibrosis, I can't speak for the other data set, but for ours, our cluster 1 really tends to express genes that characterize, an activated phenotype. And so in our data set, we're really zeroing in on really one cluster being the most activated and potentially most pathogenic. And so it'll be interesting to see how, our further verification of these clusters and potential localization in the heart, how this might impact overall cardiac remodeling.
Okay, so a couple of technical questions for you, Alessandra. So when fibroblasts were plated, what substrate did you use? And the second question is, and how long were the fibroblasts were plated?
Yes, so we plated on plastic, and they were plated for 3 days. And we are well aware of the activating, that plastic can activate the fibroblasts. And so knowing this, as we look at our data set, it's almost interesting to see, especially with the impact of our treatment, the fact that, so we recognize this as a potential limitation, but also the fact that plastic can potentially activate our cells. It may also help pull out the differences between our treated and control cells as well. So yes, we plated them for 3 days. They did not grow to complete confluence, but they were plated on plastic just to slightly expand the population for the sequencing.
Thank you. We're going to jump back over to Deepshika. What effect does BCAA have on the respiration capacity of the cardiomyocytes?
We measured the effect of BCKAs on the respiration capacity by seahorse, and we did find that it suppresses the basal as well as FCCP-induced maximal respiration rates. And it also resulted in reduced ATP production within the cardiomyocytes.
Okay, so the next question is for me. So thank you, June. This is ECI ISHR banner. But no, it's not from the big TV screen. Due to intellectual property, I cannot disclose the exact mechanism. But jokes apart, next question is for Natasha actually. And so the question is TRIM72 specifically expressed in cardiomyocyte or other heart cell as well? How do you think, that second part of the question is, how do you think the nitrosylation at cysteine 144 affect the other cells such as fibroblast? Third question, does cysteine 144 nitrosylation affect ROS generation?
Okay, so first, TRIM72 has been reported by some recent papers to affect fibroblasts, so it seems to be expressed in cardiac fibroblasts. I don't know how nitrosylation of cysteine 144 would specifically affect fibroblasts. What I can say is that there was a cell culture experiment that was published in Mark Kor's paper a few years ago before this study was done. And it also showed that this specific type of mutation, where you get cysteine 144 mutated to serine, protected against hydrogen peroxide-induced cell death. So, I mean, I would expect that S-nitrosylation would also protect against other types of cell death. And then as far as the effect on ROS generation, I don't know. I didn't look at that, but I would expect that by protecting against further injury, you would keep there from being as much ROS generation, but I didn't test that. Okay.
Great. And the last question for you, Natasha, from Jader is the specific mutation in TRIM72 affecting mitochondrial function during ischemia reperfusion injury?
Yeah, I would love to know the answer to that question, but I did not look at that during ischemia reperfusion injury. And again, I would kind of expect that by protecting against further injury, the mitochondria would be overall healthier compared to the wild type mice, but I don't know that that would be a specific function of the TRIM72 mutation versus a byproduct of the protection.
Thank you, everyone. We've reached the end of our questions, and so I think we'll go ahead and wrap up at this time. We just want to thank one last time the three presenters that we had today. Thank you so much for participating in this webinar and for sharing your science with us. We really appreciate you being here today. And one more plug for the other webinars that we have ongoing this week. Please check them out. We have some great science that's going on here. So thank you for joining us today.
Thank you.
Thank you. I mean, if you don't mind, I would just like to personally thank the three of you. Dipshika, Alexandra, Natasha, great stuff, great webinars. You were a bit of a kind of, to some extent, guinea pigs, because this was the ECI-MCI first webinar, and I think it went really well. We had great numbers of attendees, and I think the Q&A session was fantastic. And lastly, I'd like to thank Sam and Sarah. Great sharing. I mean, they've constructed this, by the way, this ECI-MCI session, and I'd like to thank them because I think this is really needed, as on top of obviously, you know, the other webinars that we've had. And we've got 2 more coming up as well. Is that right?
Yes,
Yeah.
there are 2 more. Yeah.
Yep.
So please come and join us, everyone. So thank you once again.
Thanks, guys. Have a wonderful day.